I want to start out by saying that I was honored to be invited to make this presentation. And I also wanted to say that I really appreciate the PubMLST resource. It has really helped me with a lot of my research, and I am thankful to all the staff that put together PubMLST. And I also fondly remember Allison Cody. She was always so very nice and very patiently helpful to me, and I really appreciated that. When Fran asked me to make a presentation, I was just finishing up a project, and that's what I'm going to talk about today. So I'm going to talk about the comparison of temporally and geospatially related populations of Campylobacter jejuni from chickens and from environmental waters. The objective for this study, we know that the population of Campylobacter jejuni from chickens is different from those of environmental waters. We hypothesize that analyses of the whole genome sequence will show specific loci that show selection and may correlate to colonization ability. Now, as it happens, I work in Athens, Georgia, which is indicated by this red dot on this map and this red congregation in this map. This is chicken country. The yellow land on this land use map, the yellow indicates agricultural lands, and much of the land in this area around Athens is used for poultry production. Is the major agricultural industry in the area. There is also cattle and other agricultural businesses which also show up in the yellow, but a great deal of this area is poultry. And if you look at this map, the one on your right, this has a rough topography, and this area right in here shows the valley of the Broad River, and the river samples that we took came from the South Fork of the Broad River, which is indicated in this valley right here. It happens that in the town of Athens, there is a slaughter plant that we have had agreements with, and we have been able to take samples on a regular basis. In the years 2012 and 2013, we visited the slaughter plant weekly. Each time we visited, one chicken was taken. On a Monday, we would visit and recover one chicken for isolation of Campylobacter, and on Tuesday, that would be repeated. And over the course of these 2 years, we got 67 isolates of Campylobacter jejuni. At almost the same time, we were doing recoveries from the South Fork of the Broad River, and in that time, we got 39 isolates of Campylobacter jejuni from the river. And then we subjected them to whole genome sequencing, put that into PubMLST, and got analyses of the core genome MLST. And this tree was the product of that analysis. Right off the bat, you can see that the river isolates, which have this blue coating, are largely separated from the chicken isolates that have this yellow coating. We can see if we study the tree and the outlay of the isolates, we can see that there's at least three populations right off the bat. First population includes most of the river isolates. A second population that is coded in green here includes both river isolates and some chicken isolates. And then the bulk of the chicken isolates are here. Now, these two guys here, I'm going to show you a zoom in on those two guys so that you can see that these two guys are actually more closely related to everybody down here than they are to the river isolates. This group here I separated into two different populations, which I call the chicken out group and the environmental water out group. I included this one as part of that group, even though it is distinctly a part of the chicken out group population. But the whole purpose, again, is to study the differences between the environmental isolates and the chicken isolates. And we will also talk about this exception later. To test if specific loci show signs of selection, a locus by locus FST analysis comparing the four populations, that is the chicken, the water, the chicken out group and the water out group, was used using the program Harlequin. So Harlequin does an AMOVA analysis that calculates the FST and compares different populations and gives you a p-value. When it comes to FST, it's really the p-value that counts rather than the size, the actual size of the FST. So what I have done here is created a table that shows the FST for each of the loci listed on the left of the different population comparisons. And the ones that count are the chicken and water populations, the chicken and chicken out group, the chicken and water out group, the water and chicken out group, the water and water out group, and the chicken out group against the water out group. And then the p-values are all listed on the table below. And whenever the value is less than 0.05, that indicates significant differentiation between the different populations. Many fewer of the values were not significant than there were significant. So I've highlighted the non-significant ones with a red background so that they stand out. And if we go through this table, which is rather large, by far the majority of the cells of the comparisons are significantly differentiated. If we scroll all the way down to the bottom, I have put together a table which summarizes the table as a whole. And so we have a frequency of each value and the number of times where the p-value was 0 or 0.05. These two would be the significant ones. And you can see that by far the majority of the values in all the columns are significant. And then from 0.1 on up to 1 are non-significant values. And when it comes to the Harlequin program, it kind of gives you an anomalous result when all of the members of one of the populations are being considered. When they're all monomorphic, it gives you an anomalous result. And so I changed those to a 10. So like this one here has a value of 10. That means that one of these two groups had all of the alleles were the same. You can see down here, this is a summary of the number of differentiating loci in that group. So as many as 98% of the loci are differentiating. So this is way too many to be able to pick which ones are the significant ones to determine what is giving it an advantage in the water versus in the chickens. So I started a process of subtraction. So I took out all of the loci that differentiate the chickens and the chicken out group, because differentiating the chickens from other chickens is not an important part of the study. That reduced the number of loci to look at down to about 129, and we still have no clear evidence of what makes a chicken versus a water. After that, I took out all of those that differentiate the water and the other waters, the water out group. We get a reduced table that this reduces it down to 21 different loci. So we're getting close to what we can handle. After that, I took out those that do not differentiate the chicken and the water, and that leaves this much smaller table. At this point, if we wanted to keep going, we would take out all of the ones that do not differentiate the cog and the wog and the water and the cog. The ones that are important at this point are only these three: this one, this one, and this one. These are the only three that are left that differentiate the chicken from the water isolates. And one of them is the 30S ribosomal protein, and I cannot imagine any selective pressure that that could convey for chicken. Another one is a putative molybdopterin guanine dinucleotide biosynthesis protein, and I don't know what kind of selective pressure that could have. The only one that it has of any interest at all is this N-acetyltransferase specific for PSEC product. And that happens to be a component that is necessary for for glycosylation of the flagellum, at least in Helicobacter, it's been identified in glycosylating flagellum. And so there, you could imagine that there's a selective component for colonizing chickens in that case. So when I looked at that table of the FST values, I had to, with so many of them, so many of the loci differentiating the different populations, I had to wonder if we were dealing with different species or subspecies. I will say that all of the isolates were speciated as C. jejuni based on the ribosomal protein profiles. So are we looking at subspecies? So for that, I looked to using the NAP-A sequence, nitrate reductase, which was a locus characteristic of C. jejuni subspecies Dolii, described by Miller et al., using the genus classifier. And in our populations, the isolates that you see coded in red were found to have the characteristic NAP gene loci of C. jejuni Dolii. This one, this one, this one, and this one. So these two are on the same leaf, but these two are quite different. So the Dolii-like isolates are all over the place, which actually does not match well with the description by Miller that this marker is quite distinctive and differentiating. But the markers were found in various places, and all of the other isolates had a NAP loci that was characteristic of C. jejuni subspecies jejuni. So the conclusion is that the phylogenetic analysis shows that there's probable genetic drift along with long-term population isolation. That is not to say that the populations weren't close together, because they were. Going back to this, guys that were outliers, as I say, we have one chicken here amongst the river and one river guy amongst the chickens. So they do get very close together, these populations, but they don't share any genes by recombination or otherwise. And that's what I have to say. If there's any questions, thank you.